video 2 of section 1.1 of cell biology. Okay, so we're still in that section of 1.1 of your study guide, the introduction of cells, but this is your second video. And in here, we're going to start looking at the surface area to volume ratio of cells and really looking at the size of cells and how comes they are so small. Okay, so why are cells so small? Well, it's down to efficiency. The more surface area available, the more that cells can exchange their metabolic substances across their cell membranes quickly. Okay, now it's all down to this surface area to volume ratio. So if you've got a big particle, the surface area is around the edge there. Okay, so that's the only surface area that you've got. Now, if you break that particle up smaller, you've now got all these extra surface areas that are within the initial large particle. Okay, so there's more surface area. Break it even smaller, and you've got even more surface areas. Now, that's really, really important in terms of those metabolic substances being able to exchange across the walls of cells. So cells have a small surface area to volume ratio, and there's an important reason for that. Okay, so this is just another graphic to help you get your head around the idea of the bigger shape having a lower surface area, okay, than the smaller shape. Alrighty, so how does this surface area to volume ratio affect our cells? Well, the metabolic rate of a cell is actually proportional to the volume of the cell itself. Okay, so the number of chemical reactions that will take place within the cytoplasm is larger if the cell is larger. Now, if you have a bigger cell, it needs more reactants in the cytoplasm for these reactions to take place, which means that amount of reactant has to be able to pass over the cell wall. Now, the rate at which the substances cross the cell wall depends on its surface area. So how much space is there on the cell wall or on the cell membrane for reactants to pass across? And you've got to remember that later in the unit we'll look at how these reactants pass across. Some simply diffuse, others require special methods to get through the cell membrane or cell wall. So for the metabolism to continue, substances used in the reactions have got to get in there and the waste products have got to get out of the cells. Okay, now these substances move in and out of the cell across our plasma membranes. As I just said, there's different ways they move across there. So if the surface area to volume ratio is too small, then our substances will not be able to get in and out of the cell as quickly as possible, and therefore we're going to have products accumulating inside and outside the cell, and it's not going to be efficient. Okay, surface area to volume ratio is also important in relation to heat production and loss. If the ratio is too small, then our cells may overheat because the metabolism produces heat faster than it can be lost from the surface of our cell, and if that occurs, we're going to see some cells exploding. Alrighty, so in biology, what are some adaptations to small surface area to volume ratios? So how have some organisms adapted to this? Well, the relative surface area to volume ratio decreases the bigger an organism gets. So an elephant has a smaller surface area to volume ratio than a mouse would. Therefore, organisms and cells develop strategies to cope with this problem. For example, our plant cells have a large central vacuole. So you can see the vacuole in the center of the cell there. Our intestinal cells, they have macrovilli to increase their surface area. So you see the microvilli on the end of the cell there, the little finger-like structures, projections. So that increases their surface area. The leaf palisade cells, well, they're column shape, okay, and that is to, again, increase that surface area. Also remembering that they have the vacuole inside as well. And then for example, our elephants and our desert foxes, they have large ears, okay, and that is to try and increase their surface area volume ratio. Whereas polar bears that want to reduce the amount of heat they lose, they have small ears. The wall of our intestines, to increase the surface area there, they have what we call villi. Okay, so these finger-like projections in the wall of the small intestines. Okay, how do we see cells? Okay, and you'll all know that to see small things in a science lab, we use some kind of microscope. Here we've got our microscope with all of its parts labelled. It is important that you know the parts of a microscope. When we're doing microscopy in the lab, which we will be doing this week, you need to know what the different parts of the microscope are called when you are using the equipment. Okay, so when we're looking at items that are very small, we have a whole scale of SI units that we use, okay? Now you need to know some of these relative units, okay? So if we start at 1 meter and we reduce our size by 1,000, okay, we're reducing it by 10 to the negative 3, okay? So we're removing the 3 zeros and negative 3, okay? So 10 to the negative 3 of a meter is the equivalent to a millimeter. 10 to the negative 6 of a meter, or 10 to the negative 3 of a millimeter, equals what we call a micrometer, its unit being UM. Okay, 10 to the negative 9 of a meter, or 10 to the negative 6 of a micrometer, or 10 to the negative 3 of a micrometer is 10 to the negative 6 of a millimeter, or 10 to the negative 3 of a micrometer is a nanometer. So we'll do some more practice in class around those measurements. It takes a little bit to get your head around them. Just have a read through and we'll start to get that understanding. Now, if we're looking at relative sizes, we've got some different items here that you would know of, different organelles, some organisms and some structures that you would have heard of. Okay, and we've got their relative sizes there. So now we're looking at the smallest parts of our universe, and we'll go right through to the biggest parts of our universe. Okay, so when it comes to calculating magnification, all right, when you're given the size of an image and the actual size of the organism, so what we need to do, our magnification is the size of the image divided by the actual size of the specimen. Okay, now we can use one of our little triangles for that. So we've got our magnification, our size of the image, and our actual size of the specimen. Okay, now if we wanted to find our magnification, we divide the size of the image by the actual size. If we wanted to find the actual size, we'd divide the size of the image by the magnification. So this is our divide line. If we wanted to find the size of the image, we'd do the magnification times the actual size. Okay, so let's practice this. We're told that our image is 5 millimeters in length and the actual size of our Daphnia is 25 micrometers. Okay, so what we've got to do first of all, we need to ensure that all the units are the same. So we're going to convert our millimeters to actual size as such. So we're going to say 5 times 1,000 gives us 5,000 micrometers. Alrighty, so our size of the image is 5,000 micrometers and our actual size of the specimen is 25 micrometers. So we're going to do 5,000 divided by 25 gives us 200 times is our magnification. Okay, so it's that 5,000 divided by 25 gives us 200 times magnification. Okay, so here we've got an example. We can see our actual size is 400 millimeters. Okay, now we're going to multiply that by our 1,000 so that we're in the same units. Okay, and then we've got 4,000 divided by our 2,000, which gives us 2 micrometers for our actual size. Okay, so remembering here we've got our size of our image and we've got our magnification. Okay, so, and we're trying to find out our actual size. So our size of our image was our 40 millimeters, so we had to convert that to be at the same level as our magnification. Okay, here we have our magnification here, an image. All right, now actual scale bar measures 15 millimeters. 15 millimeters is the equivalent to 150,000 micrometers. Okay, so we say 150,000 micrometers divided by our 50, which we've got here, gives us our 3x magnification. So we measure the actual scale bar in millimeters. We convert the units of the scale bar if we need to. And then we go actual scale divided by the stated length of the scale bar gives us our magnification. And just remembering our triangles, because our triangles are really good just to help us get our head around this. So actual size here is our image divided by our magnification. If we're looking for our magnification here, we do our image divided by our actual size. And that's all for this one, guys. We'll be doing much more practice on this in class, don't worry.